All right. This is Roy Trevelyan. I'm going solo today. My wife is gone, so we'll see how this works. Anyway, I'm going to give you a quick bio bite on mitosis and meiosis. And we're going to worry less about the stages and more about where people get hung up. When we talk about mitosis, we're talking about cell division, right? So I brought this onion that's sitting on the counter over there. You're going to see mitosis on the edge of these roots, and you're going to see mitosis here, the two parts that the plant is growing. Now, some of you guys came in lab and you did the selfie and you realized that most of our cells are not doing mitosis. When you looked at your cells under the microscope, your cheek cells, you realized that 90% of our cells are not doing mitosis. The DNA was all packed together. Remember, we talked about 6 feet of DNA in that nucleus, and we just called it chromatin. Now, when we look at cells that are dividing, like in this root cell, in some of our cells, but majority of our cells are not going to be dividing at this time. Oh, maybe our liver cell. Those are active cells that divide. But what the cells are dividing, you get what they call a chromosome. So I'm going to give you a chance to go find your pack of chromosomes, right? There should be in your kit. And you're going to need to put on your little straws with the letters on them, and those are going to represent a chromosome. So where it gets confusing now, one chromosome consists of 2 chromatids, right? And these little straws on here are representing the genes that are on the chromosomes. Now, we're not going to go crazy on the number of genes we put on these chromosomes, but realize that the human genome consists between 20 and 25,000 genes. In other words, if you could pack in 25,000 of these little straws, all with different forms of genes on them. Now remember, the way chromosomes, the way DNA controls the cell is by making proteins. Proteins, remember, are things like enzymes. Enzymes lower the activation energy so that reactions can occur. The nucleus does control the cell. Now, the 2 different processes we're talking about today are mitosis and meiosis. Oh, wait, sorry, one more thing. This is where a lot of people get hung up. These 2 chromatids are identical. There's nothing different from this chromatid and this chromatid. So in mitosis, we're going to pull these 2 chromatids apart, and we're going to still call them a chromosome. And the reason we can do that is if I have one, I can always make an extra copy, right? Whatever's on here, DNA is a miracle because it can self-replicate. It can make an entire new copy of this chromosome. So where people get hung up is when I pull this chromosome apart into 2 chromatids, this one's going to go into one cell, this one's going to go into the other cell, but the chromosome number hasn't changed. When we talk about mitosis, we're going to talk about mitose. I don't remember what meiosis does. I only know that mitosis does mitose. Toes are body cells. And body cells are going to go from diploid to diploid. So we're not going to talk about chromosome number in terms of an actual number. We're just going to use this term diploid. Diploid means a full set of chromosomes. Now, I'm going to cut to the chase because everybody gets this wrong. We have 46 chromosomes. So we have 46 of these X's in every one of our cells, all packed together inside there, right? And we have 23 pairs. So 2 of them would be orange. You know, I got some down here that you can see that are red and some are purple. Here's one that's green. So imagine you got 23 pairs or a total of 46 chromosomes. Now, when we look at meiosis, when we talk about sex cells, right? I don't remember that all my cells in my body do mitosis except one different type of cell, and those are the sex cells. Sex cells, the fancy name for these are called gametes, and gametes are sperm and egg. Now, if we think about it logically, I hope, when we go to make sex cells, we're not going to stay at diploid. I don't want a full set of chromosomes. I only want a half a set of chromosomes. So we're going to go from diploid to haploid. Almost sounds like half, doesn't it? We're going to go from 46 in humans to 23 in our sex cells. So all of our cells will contain 46, but in humans, our sex cells, our gametes, are going to contain 23. Right? Now that should make sense to you because when the sperm and egg come back together, we want to be back at 46. So in all organisms, when we do meiosis, when they want to reproduce, they're going to go from diploid to haploid. But in mitosis, we're going to stay diploid to diploid. And again, in mitosis, it's as simple as this. All 46 chromosomes are going to line up and they're going to pull apart. Remember, when I pull these two chromatids apart, the chromosome number has not changed. As long as I got one chromatid, I can always go and make another copy. So all 46 are going to line up single file, and we're going to pull them apart, going from diploid to diploid. If I'm a human being, 46 to 46. If I'm a corn plant, we're going to go from 10 to 10. Chromosome number doesn't change. Now, we were really disappointed to find out, kind of took a notch out of how cool we thought as humans. These genes on these chromosomes, there is only an estimated 25,000 genes. We thought for sure, as complicated as we are, there should be a lot more genes, but that's all that makes up humans. Now there's some reasons for that. We'll get to those later. But anyway, I want you to remember, if you could remember this when we do mitosis and meiosis, if we pull the chromatids apart, does the chromosome number change? And the answer is no. We go from diploid to diploid. So we're going to stay away from the numbers because we're going to talk about different types of organisms. We want to use these adjectives, diploid to diploid. These types of cells are called somatic cells, body cells. If I yank any cell out of my body, it should be at 46. The only place we're going to see diploid to haploid, to half the number of chromosomes, is in our sex cells. All right. If you can drop down to the bottom now, I want you to take a look at the two sets of chromosomes I got on here. And again, you're going to go find your kits. You're going to have to find those pipe cleaners, and you're going to have to put the genes on them, okay? I tried to put different genes on the different chromosomes, and I just want to show you a couple things here before we're done. Again, we're not going to talk through the processes. But if these are my chromosomes, let's pretend this is an organism. I got 2 reds, 2 purple. My diploid number for this particular organism would be 4. My haploid number, when I go to make sex cells, will be half of that. Well, let's go ahead and do meiosis very simply. I take one of the reds, I take one of the purples, and now I only got 2 in my cell. That's a sex cell. I went from diploid, 4, to haploid, 2. If this was a human being, remember, we'd have 46 chromosomes, 23 pairs, and I would go and yank 1 out of each. Now, a couple things I want to talk about here quickly is that if you look at these purple chromosomes and the red chromosomes, they both have genes, but they're located at different spots. The fancy word for that is loci. The blue and the yellow genes are separated almost completely, one on either end of these chromosomes, where the purple ones, these genes are really, really close together. Now, the technical word for going to use that is for linked. So what I did now is I just put letters up here to show you what's on these straws that you can't see. But you're going to take these and you're going to do mitosis and meiosis after we talk about them. So you need to get your pipe cleaners. I'm going to steal a lesson from Mr. Denny, and I'm going to have you take all 8 of your chromosomes and you're going to video mitosis and meiosis. The short version, like 5 minutes long. That's it. Remember, talking the talk helps. Anyway, on the red one, I got big A, and on the other red one, I got little a, little a. Later on, we're going to call this heterozygous. I got this chromosome from mom, this chromosome from dad. Dad gave me big A's, mom gave me little a's. And the same is true down here for the L's. The yellow represents the L's. Dad gave me big L, mom gave me little l, little l. Now, these genes that are on this same chromosome are over here. I got big T, little t, little t. I got big R, representing that lime green, and little r over here. But they don't have to be. Mom could have gave me big A, and Dad could have gave me big A. A term we're going to use later on that's homozygous. So in meiosis, remember, all we're going to do is take one of the purples and one of the reds, and we're going to make a sex cell. In mitosis, what we're doing is we're pulling the actual chromatids apart. And remember, when you pull these chromatids apart, the chromosome number doesn't change. The only time the chromosome number is going to change is when I take one of these homologous pairs. What I mean by that is the two chromosomes that look alike. They're the same size, the same shape, and they carry information about the same traits. And only in meiosis do you find your homologous pairs. Only in meiosis, when we go to make sex cells, gametes, do you line up 2 by 2. In mitosis, when you make your video, never show your homologous pairs together. Only in meiosis. And the reason we have to do this is so that we can take one of each. Remember, we're going to go from diploid to haploid. Lastly, I want to show you something on these genes here. These genes here, we are going to say that are linked. Sometimes what happens is the chromosomes will get close enough and actually tangle up. Now, let's see if I can do this on the video. But I'm literally going to flip this big A with this little a. They switched places. It's a physical entanglement of these chromosomes. Now what that's going to, okay, pretend I put that on there. What that's going to look like up here is, sorry, not big A, I meant big T. I'm going to flip this T with this little t. In other words, some of the genes from one of the chromosomes is going to switch places. So now this cell, this sex cell, is going to get a little t, and this sex cell is going to get a big T. It's a unique property that we're going to call crossing over later on. Now again, this is just to give you some ideas. And the things you want to remember with the remaining time I have is this. When you pull the chromatids apart, well, go get your pipe cleaners too. I'm warning you to do that because we're going to start talking about this. When you pull the chromatids apart, chromosome number does not change, right? I know that's weird, but one chromatid is equal to one chromosome. That's where everybody gets hung up, right? If I have one copy, I can always make another copy. Each cell gets a copy. We go from diploid to diploid. Only in meiosis, sex cells, am I going to take one of each pair. All right, so now I'm going to give you a diploid number. This will be the last thing I do here. I'm going to give you a diploid number of 6. 1, 2, 3, 4, 5, 6. Different organism, right? Again, if this was humans, we'd have 46 chromosomes out here, 23 pairs. And what I'm going to ask you to do in your video is to show me, you don't have to have your face on, you can just use your chromosomes. But what I want you to show me in meiosis is I want you to show me going from diploid to haploid. So I want you to take one of the oranges, one of the reds, and one of the purples. Now, what's the chromosome number? Well, it's no longer diploid, it's not 6, it's haploid, 3. Lastly, when we go to make sex cells out of this is going to be a sperm cell, for example. Realize something. This has got a big A and this has got a big A. This has got whatever letter that is, and that's got the same letter. I don't really need to keep those copies. Just like in mitosis, meiosis 2, this second division, we're going to pull all of these chromatids apart. Right? Now remember, when you pull chromatids apart, it does not change the chromosome number. Right? We're going to still be at haploid for this sex cell. Oh, there goes my genes. By the way, I used a little twist tie to get those chromatids to stick together. It's called a centromere. Anyway, look at what we got now. We got one cell at haploid. Remember, we were at, oh, what did I do there? Sorry, we got 2 cells. Too early in the morning here. 2 cells, right? One at haploid, one at haploid, right? We got one purple, one orange, one red, one purple, one orange, one red. The fact of the matter is, when we do meiosis, when we do sex cells, because there's copies and we do meiosis twice, we're going to end up with 4 cells with half the number of chromosomes. Notice the diploid number again was 6, right? We had red ones, purple ones, and orange ones. And now we got one of each, right? Not any half, but one of each. And we're at the haploid number. I'm Roy Treveline. This is Biology Bites. Thank you for listening.